Pollination is the transfer of pollen grains from the anther to the stigma of a flower. After the pollen grain is transferred to the stigma, the pistil recognizes whether the pollen grain is of the right type, which means that it must be compatible for fertilization. This requires that the pollen grain must belong to the same species and must not be self-incompatible if from the same plant. The chemical substances present in the pollen and the carpellary tissue interact with each other. If the pollen grain belongs to the same species, the carpellary tissue recognizes it and triggers the formation of the pollen tube. Recent studies suggest that non-self recognition of the pollen grain involves various proteins and peptide molecules that bring about pollen-pistil interaction. Once the pollen grain is accepted, the sticky stigmatic secretion containing sugars and organic acids helps the pollen grain germinate. On absorbing the stigmatic secretions, the pollen grain is hydrated. It swells and ruptures at the thinly placed exene of the germ pore to form the germ tube. The tube nucleus and the generative nucleus enter the pollen tube, as the pollen tube makes its way through the long style of the pistil. The tube nucleus lies at the tip of the growing pollen tube and is responsible for the growth of the pollen tube. The generative nucleus divides mitotically to form the two male gametes. The pollen tube grows through the style and reaches the ovule, located within the ovary. The tube nucleus degenerates and the two male gametes enter the embryo sac of the ovule. The filiform apparatus in the synergids guides the entry of the pollen tube into the synergid. The pollen tube enters either of the synergids and releases the two male gametes into its cytoplasm. The entry of the pollen tube through the micropyle is called porogamy. Other modes of entry are the chalazogamy, in which the pollen tube enters through the chalazal end of the ovule, and mesogamy, in which the pollen tube enters through the integuments. Inside the embryo sac, one of the two male gametes fuses with the egg cell to form the diploid zygote. Fusion of the male gamete with the egg cell is called syngamy. The other male gamete fuses with the two polar nuclei in the central cell to form a triploid nucleus, called the primary endosperm nucleus. The formation of the primary endosperm nucleus involves the fusion of three haploid nuclei and is thus known as triple fusion. The process of fertilization, which involves the fusion of the male gamete with the egg cell and the fusion of another male gamete with the polar nuclei, is called double fertilization. Double fertilization involving two fusions is a feature that is unique to angiosperms. The process of double fertilization was discovered by a Russian biologist, Sergei Navashin, in the year 1898. The primary endosperm nucleus, formed by the fusion of the male gamete and the two polar nuclei, divides to form the endosperm. After fertilization, the zygote, formed by the fusion of the male gamete and the egg cell, divides mitotically to form the embryo. The endosperm, consisting mainly of starch, provides nutrition to the developing embryo. The formation of the endosperm is a unique feature of double fertilization and is thus seen only in angiosperms. After fertilization, the ovule forms the seed, and the ovary enlarges to form the fruit.